The last way in which DNA can be transferred horizontally is using something called bacteriophage. These are bacterial viruses. They only infect viruses, or only infect bacteria. And here is a picture of what looks like, at least one of them. You've got DNA in a head on top, surrounded by a protein coat. This is a slice through, and that's a 3D model. This region here forms what essentially is a syringe. They can use to inject the DNA into a bacteria. The tail fibers that are on the side here are used to recognize that it's the right kind of bacteria. They're very specific. So they bind to the bacteria. Bacteriophages are crazy common. Scientists estimate there are 10 to the 30th bacteria in the world, but they also estimate there are 10 times that number of phage or bacteriophage. And we have very little knowledge about the diversity of phage. So how this works is as follows. A bacteriophage finds a bacteria, it attaches to it, it then injects the DNA into the cell. That DNA is then going to be copied. You get lots of copies. You then are going to start to make new proteins from that DNA to make new phages. During this process, the host chromosome is often broken up into pieces, the DNA. And then finally, you've got new phages inside the cell, and proteins are made that cause the cell then to explode. And the phages can go out and infect another bacteria. A typical phage might produce 100 new phage in just 20 minutes. So this is extremely effective. All right, if you want to see what these look like in the lab, it's shown here. So on this plate, again, you have a layer of just bacteria growing here. It's all cloudy. What you can see are little holes, what look like little holes. And those tiny holes are where a phage was infected and killed all the bacteria in that area. Now, the reason I'm telling you about this, there are two reasons. The first reason is that these can spread antibiotic resistance by a mechanism called transduction. Transduction works like this. If you have a cell and it infects, you have a phage and it infects a cell, and I've made it different color DNA. It's not really different colored, it's just for making it easy. The phage has blue DNA, the cell has red DNA. It infects, it produces new phage. Sometimes when the phage is being assembled in the cell, it makes a mistake and it will package a piece of chromosomal DNA. That piece of chromosomal DNA could include an antibiotic resistance gene. So that when this red phage infects the cell, it isn't going to kill the cell because it doesn't have phage DNA, it has E. coli or bacterial DNA. And it can recombine in the chromosome and add those genes to the bacteria. So these can transfer antibiotic resistance. But I want to emphasize the most common way is conjugation. To end today, I'm going to talk about alternative approaches to antibiotic resistance. We're going to talk much more about this later, but there are two things that are connected to what I've been talking about tonight. So the first one is what you just said, the idea of phage therapy. Why not just use phage to kill bacteria? That's their job, kind of. So this was tested around the time of the discovery of phage in the 1920s. They had that idea, but they got very inconsistent results. It worked sometimes, it didn't work sometimes. We now know that not every phage infects every bacteria. So they didn't know that at that time. So they thought the phage could kill every infection. But in fact, it's very specific. So it didn't work. And then antibiotics came around and said, who cares? Except in certain places in the former Soviet Union, where it continued to be studied. And so there is a big center in Georgia, which has been studying phage therapy for the last, I don't know exactly, 70 years or so. But now, in the West, this idea is being revitalized. So there is a lot of talk about using phage to kill infections. And to date, there are still in clinical trials, so we don't know how effective it's going to be. But there are cases where it's being used on patients that have no other treatment. And what are the examples I'm going to show you now? So there was a patient who had a bacteria which had one of these efflux pumps that I mentioned before. This was a patient who had an infection in his chest he had had some heart surgery. And he had an infection that was completely resistant to everything, primarily because of this efflux pump. It was pumping out the antibiotics. So the researchers found a phage that could bind the efflux pump and infect this cell. All right, so what happens there? I'll show you. And this goes to what you were saying as well. So here is the original strain. It's got an efflux pump. So it's antibiotic resistant, but it's sensitive to the phage because the phage binds the efflux pump. If it mutates, which bacteria do, so they become so that it no longer is sensitive to the phage, it means it loses the efflux pump. And now it's sensitive to antibiotic, but resistant to the phage. If it mutates again, it's going to go back to this situation. So what they were able to do is treat the patient with both phage and the antibiotic at the same time. And this worked. The patient recovered from the infection. So using both of these together might be a very effective way to do this. But this is all very new. This was published, I think, a year and a half ago or so. A second way you can think about efflux pumps in particular, I want to mention. Another approach people are taking is, so here's the efflux pump pumping out antibiotics. Instead of worrying about the resistance in general, why not just find a molecule that could block the efflux pump? And there have been cases of finding inhibitors, still early clinical testing, that actually just bind the efflux pump, which prevents the antibiotic from being pumped out. That's another type of approach that's being taken. So to summarize what I've talked about in this last section, we've been talking about the spread of resistant bacteria. And you can look at this slide when you get home. Next week, we're going to talk about another aspect of this, which has to do with the environment and use in livestock. Because antibiotic resistance doesn't just happen in people, it happens in the environment as well. So we're going to talk about that next week. And then lastly, is what I mentioned, if you really want to learn about microbiology news, there is a Facebook and Twitter account that I run that you can follow if you like.